Welcome back, everyone, to the second day of the HIBALL Winter School. In this session, you'll learn about multimodal image integration of BigBrain with other data modalities, and specifically the BigBrain Warp software. And for this, it's my pleasure to introduce Dr. Casey Paquola, and she carried out postdoctoral studies in my lab at the MNI and has recently moved to Jülich to stay in the HIBALL family. During the time in the lab, Casey has made very important contributions to study microstructural differentiation of both human allocortical as well as neocortical regions, and to explore associations between microstructure gradients in the human brain and large-scale functional networks. And today she'll present BigBrain Warp, which is a wrapper tool to give helpful functionalities to integrate BigBrain with other data modalities, specifically structural, microstructural, and functional MRI. Casey, please take it away.
Thanks, Boris. Okay. Just getting my, there we go. Cool, can you see the title page, Boris?
Yeah, perfect.
Awesome. Okay, so good morning, everyone. Thank you, Boris, for the introduction, and also thank you to Susanna and PJ for organizing all of this. It's a lot of hard work, but I think it's been really enjoyable so far. So Boris already said that I've just recently moved from Montreal to Jülich, and so together with these two labs, we've been working to build BigBrain Warp. So this is a toolbox for multimodal integration of BigBrain. So in the first half of the talk, I'd like to discuss kind of the conceptual backgrounds and motivations for this toolbox, especially focused on the relationships between cytoarchitecture and function. And then in the second half, we'll really delve into the toolbox itself, and I'll show you more a demonstration of how you can use it. Okay. So in vivo neuroimaging, I think, has allowed us to really comprehensively characterize the macro-scale landscape of the human brain. So for example, you see a functional MRI here, and it enables this visualization and analysis of ongoing fluctuations in cortical activity. So I think this video epitomizes the balance that we strike with the vivo imaging between a spatial and a temporal resolution. And this has profoundly changed our understanding of the dynamics of the human brain that occur from second to second, but also across the lifespan and even across evolution. And this stems from this major advantage of MRI that we can roll it out in large cohorts of individuals to assess the similarities and differences. And with that, it enables this examination of the coordinated functions across the brain. I'm not looking at the chat, by the way, so Boris, just feel free to interrupt me if there's anything that I should know about. So inspecting the functional connectivity has led to these new depictions of cortical organization. The neuroimaging field has really embraced some of these conceptions, such as clustering of the cortex into communities and networks. That's what you see on the top here, the famous Junio and Krenen networks, or also looking at the principal gradient of functional differentiation, such as this one by McGill et al. And these canonical examples demonstrate how complex patterns of cortical activity can be deconstructed, and then they become a useful framework in which to understand other variations across the cortical mantle, such as neurobiological properties or individual differences. And so BigBrain is helping us to uncover one of these regional variations, specifically the composition of cells, like you can see here. I think it'd be really helpful for us to have a flexible machinery to relate cytoarchitecture and function, because at the moment, their relationship is, well, we know it's very complicated, but it's also quite unresolved what their relationship is. There are a couple of linking principles that were primarily developed in the 1990s related to both cytoarchitecture and function having common axes, or that you can predict connectivity from cytoarchitectural similarity. But whole cortex analysis of these principles in humans was only really made possible, I think, by BigBrain. So I'd like to start off by discussing how BigBrain MRI analyses have helped us to test these principles. Marcel Meslin described cortical organization as a series of rings. So here you can see the outermost ring, which has primary sensory areas. And each ring inwards reflects the synaptic steps. The deeper the ring, the greater the synaptic distance from primary sensory input. He also identified that cytoarchitecture systematically varies across the rings. The outermost regions exhibit high laminar differentiation, those 6 or more cortical layers that are clearly definable. And then as you move inwards, the laminar differentiation decreases, and the final rings are then dysgranular and agranular. So combining this with primate electrophysiology, Meslam also showed that the type of processing systematically varies across these rings. So processing is increasingly integrative. So you start from more unimodal processing to heteromodal, and then some would even call these deeper layers amodal processing. So together, this kind of created this theory that there are common axes of cytoarchitecture and functional differentiation, which Meslam referred to as the sensory fugal axis. We can test this principle using BigBrain and BigBrain Warp, and we've published on this previously. So if you're interested in more details, I'll direct you to our 2018 PLOS biology paper. We characterize cytoarchitecture across BigBrain using the grade level index. In other words, we sampled the staining intensity in a straight line from the peel to white matter surface. So you have this staining intensity profile. You can see 2 of them here from 2 different regions. And so you show variations in the cellular density or cell size as you move through cortical depths. Then we looked at these across the entire cortex and did a pairwise correlation between these profiles to establish cytoarchitectural similarity across the whole cortex. And then we're interested in extracting these principal axes of cytoarchitectural variation. So we employed diffusion map embedding. It's a nonlinear manifold learning technique. It reduces the dimensionality of the matrix to a small set of eigenvectors, which are ordered according to the amount of variance they explain in that matrix. So I often refer to this as a microstructure profile covariance matrix. So we're trying to understand what are the principal axes of variation in terms of that microstructure profile covariance matrix. So the first 2 eigenvectors that we obtain from this type of approach are always an anterior to posterior gradient and a sensory fugal gradient. So very similar to what Meslam had shown in his work, this sensory fugal gradient. And that's what you see down here in the bottom left. So on the left-hand side, you can see these gradients projected back onto the big brain surface. It looks like it has little triangles all over it, because I've performed a type of mesh decimation. So big brain surface has 320,000 vertices about. So it's a little bit hard to perform pairwise correlations between all these vertices. So instead we downsample that to about 10,000 vertices, and we essentially group vertices together into little patches. So we go from a mesh of 320,000 to 10,000 in such a way that preserves the overall shape of the big brain surface. And then for the vertices that we've disregarded in that process, we essentially match them to the closest still maintained vertex. So that's why you have this cool tessellation pattern. So here you have this anterior posterior gradient, and you also have this sensory fugal gradient. So more sensory areas are in blue and more transmodal or amodal areas are in red. Then we were interested in relating this to function and especially to other forms of in vivo imaging. So we needed to move it into a standard template space. So here we're moving it to FS average 5. So this is a standard surface template that's used throughout the FreeSurfer ecosystem. And here we just used a nearest neighbor interpolation from big brain surface to FS average 5. And so you can see, let's look at a few little areas here. So if you see in this temporal parietal junction area, you have a red part on the big brain, and it really just jumps over to your FS average quite nicely there. So we can just inspect this to see that we have a good alignment of the patterns, so the colors on different sulci and gyri there. So we can transform this histological gradient now to FS average 5, so in an in vivo space. And then we can compare this to other forms of in vivo imaging. So here we have these first 2 gradients. We've employed a little bit of smoothing here as well, because we're using nearest neighbor interpolation. Then we smooth on the surface as a way to almost artificially use like a trilinear interpolation. But we can also test with and without smoothing and get quite similar results actually. So we were interested in whether we could replicate this approach with in vivo imaging, first of all, and that's what you see here in this micro G1. This was using the very similar approach to what I just showed, but instead of using BigBrain and histology, we actually used quantitative T1 imaging from just a standard MRI sequence. And so we also created these staining intensity profiles, correlated them, and then employed diffusion map embedding. And so we see quite a strong similarity between this sensory fugal gradient that we obtain in living humans compared to the postmortem data as well. And we also took the functional gradient, which I showed earlier from McGill et al., where you employ diffusion map embedding on a resting state functional connectivity matrix. And now, because these are all on this common atlas, FS average 5, we can directly correlate them. So here you can see a correlation matrix, and we see a very strong concordance of 0.64, that is, between the histological and the microstructural gradients, these sensory fugal gradients. And we also see that these are very similar to these functional gradients. So it really supports Meslin's theory on the common axis of cytoarchitectural and functional differentiation in the human cortex. And then on to another principle that we could test. Helen Barbas's group introduced the concept of the structural model, in that microstructural similarity predicts connectivity. They showed that in cats and macaques, that the more similar two regions are in terms of their lamination, the more connections that they share. And for example, using neuronal density as a proxy for cytoarchitectural type, Boole and Hilgertag clearly demonstrated this relationship in the macaque cortex. You can see this plot on the left here, where they showed that the combination of distance and density, so density is their proxy for cytoarchitecture here, you can get a really solid prediction of the existence of projections. And the structural model also makes claims on how structural type predicts the laminar origin of projections. So dissimilar cortical areas project to distinct layers, so such as agranule and eulaminate 2 here. So you can see they're kind of jumping between different layers where they terminate. Whereas the laminar origin of projections is more similar when you go between more similar types of cytoarchitecture, like dysgranular to eulaminate 1, which are more similar. And this also relates to feed forward and feedback connectivity. So it's a really good marker of whether something is feed forward or feedback. So deeper origins relate to feedback, and shallower origins relate to feed forward. And this can be clearly seen in an example of agranular to eulaminate. For example, if you're going from agranular to eulaminate, it's more likely feedback, whereas eulaminate to agranular is more likely feed forward. We're really interested in this idea. I wanted to test it in humans, and specifically wanted to test it in the mesiotemporal lobe, where there's so much variation in the cytoarchitecture, and obviously this really beautiful curled up section of the cortex. So the isocortex, which you can see here there's a purple line there. So this is from a BigBrain coronal section. So there was already existing these isocortical models from the original release of BigBrain. And then there was also recent work by Jordan de Cracker and Ali Khan's lab at Western. So they created this really awesome model of the hippocampus in BigBrain. So they manually segmented the hippocampus, and then they created this internal coordinate system within the hippocampus. So you have 3 dimensions, one that's from inner to outer, which is very similar to peel to white matter. Then they also have anterior posterior, and then proximity to the isocortex. So it's actually, this is the most proximal, and then this would be the most deep in at the dentate gyrus. So this really helped us because we could then link these 2 different models of the isocortex with the hippocampus. And that's what you see here in the center, where we've linked these 2 models and basically got rid of all the superfluous things where you typically jump over the hippocampus in these cortical surface models. So now we have a continuous surface model of the mesiotemporal lobe that includes both isocortex and allocortex, where the hippocampus is the allocortex. And then using this, we could create this geodesic distance to the intersection. And we use this as a proxy of this graded transition from iso to allocortex, because there was also a lot of research showing that there's cytoarchitectural variation that's gradual from the isocortex. So from kind of fusiform, parahippocampal gyrus, entorhinal cortex, then you have subiculum, CA1, 2, 3, 4. So this whole transition is also very gradual change in the cytoarchitecture. So that's what we're looking to model here. And you can see that on the right with these staining intensity profiles, you see a gradual change in the cellular density at certain layers as you move from isocortex on the more darker side, so this area out here, as you move into more allocortex in these bright yellow colors. Then we next wanted to transform this to a space that we could perform in vivo imaging in. So we took this surface model that was in the BigBrain histological space, and we projected from surface to volume. So that's where you see here, we now have colored the voxels based on their position across this axis. So you have more isocortex into allocortex. And then we could use a volumetric transformation to go from BigBrain histological space to MNI 152, which is another standard volumetric template used in neuroimaging. So as you can see here, now we have this map that was only possible to create in the high resolution space of BigBrain, that's now in the in vivo neuroimaging space. And in one analysis, we used this to define seed regions and then calculate the effective connectivity within the mesotemporal lobe. We were particularly interested in whether signal flow followed the isoallocortical axis and whether this primarily occurred in a feed-forward or feedback direction. We found that effective connectivity decreased with greater differences along the isoallocortical axis. That's what you see here in this right plot. So this shows that the relationship between cytoarchitectural similarity with effective connectivity that aligns with the structural model. We did not observe a significant difference in this effect in the isoallocortical versus allo to isocortical direction. So you have the green line versus the purple line. They both show a very similar relationship between the cytoarchitectural differences and the strength of effective connectivity. So we saw that this relationship seemed to be preserved in a feed forward and feedback direction. And then finally, in another analysis, we sought to expand our scope to large-scale cortical dynamics. We tested whether connectivity between isocortical areas and the mesotemporal lobe was associated with the isoallocortical axis. So indeed, we found that functional connectivity of the red areas here was higher towards allocortical parts of the mesotemporal lobe, whereas the blue areas had stronger resting state functional connectivity to the more isocortical areas of the mesotemporal lobe. So I thought this was quite interesting because there'd been a lot of research on the anterior-posterior differentiation of functional connectivity from the mesotemporal lobe. And our work shows that the isoallocortical axis is also really another important dimension of this that can explain the broad range and complexity of connectivity that you see across the cortex. And I think it'd been largely overlooked in the past because we didn't have an adequate model for in vivo neuroimaging, and we really needed to recruit histological models of the mesotemporal lobe and bring them into this in vivo imaging space to elucidate this relationship. So I've shown you a couple of examples of how we're able to test and expand upon these linking principles between cytoarchitecture and function using integrated BigBrain MRI analyses. And I think this is really just the beginning of what we can uncover, and we could apply similar workflows to interrogate principles proposed in other exciting new work as well. So for example, Alex Goulos has shown that that the prediction of connectivity from cytoarchitecture also varies depending on the length of the tract. You see there's a stronger relationship here in longer tracts. And this is something that we could also extend and explore in the humans with more BigBrain MRI analyses. And in that paper, as well as other recent work by Garcia Cabezas et al. In the human frontal lobe, there's been people who have highlighted that there's more cytoarchitectural features that vary across gradients. It's not just laminar differentiation, which we've focused on so far, but also the size and the morphology of neurons. So all of which could be more comprehensively characterized, I think, with BigBrain and related to more macro scale features of in vivo imaging. And finally, the microcircuit models we currently use are primarily based on the visual cortex, so as we see here in this top plot. However, we could anticipate that the microcircuitry is quite different depending on different types of cytoarchitecture. So you here see a very neat proposal by Bull and Hilgetag. So you see in a granular cortex, the type of microcircuitry involved could look very different. So I think this shows there's a really great opportunity that we could have to use cytoarchitecture to inform these functional models as well. So that's where BigBrain Walk will hopefully come in. We have created this toolbox as an ecosystem for integrative cytoarchitectural analysis. On one side, it's hopefully going to be a place to facilitate more cytoarchitectural characterization. And on the other side, it will take in macro scale features of the brain and then bring them into a common space. So in BigBrain Walk, we're providing a set of tools and examples for multimodal integration of BigBrain. Both BigBrain and MRI will need to subject them to some type of pre-processing to extract these meaningful features. And then for comparison, you'll need to transform them into the same space. And finally, you'll need an analysis strategy of how to compare them. And BigBrain Warp will help to cover 3 of these bases, those in purple. We've pre-generated some cytoarchitectural features, and we try to provide lots of links to previously published work. We've also created some very simple one-liner transformation scripts, and we've written comprehensive tutorials on how to undertake such multimodal analyses. On to the cytoarchitectural features. I've been through this a little bit earlier in the talk, but I just want to point out that a really effective approach to characterize cellular architecture of the cortex is to look at the staining intensity profiles. So this technique was pioneered by Carl Zillis and Katrina Muntz, and they take the staining intensity across cortical depths. So you can imagine sampling along this purple line in B, from the peel to the white matter boundary. And you can visualize this as a plot of staining intensity by cortical depth. And there's already some really noticeable features from this, such as the peak in layer 4. So we and others have utilized this approach in BigBrain previously. I showed you a couple of examples. Also, Conrad's going to speak later today about using this to define the lamina of BigBrain. And we can use this in combination with morphometric models, such as the hippocampus or the mesotemporal confluence that I discussed before. And so we're hoping to we'll release these profiles so people can just use already extracted profiles in BigBrain Warp. We'll also cover a lot of these topics in the tutorials and provide links to these different models, so it can be a really easy go-to point for when you're starting BigBrain MRI analyses. So at the heart of all these analyses will be the transformations between BigBrain and standard neuroimaging spaces. So on the top, you have all the volumetric spaces, and on the bottom, the surface spaces. We have BigBrain original histological space here. Then we have BigBrain Sim, which is already in an MNI 152 space. It's been registered to that space already. And then you have maybe more standard atlases from vivo imaging, like MNI 152, also known as ICBM Sim, or the FS average surface. So there already exists a lot of relationships between all these different spaces of how to move between different ones, how to sample from different spaces. So we want to make this a little bit simpler for users and just kind of wrap this all up in a couple of bash scripts. So this is really the core of BigBrain Warp, is we're going to really keep all that stuff under the hood, and so people can just really simply use these scripts. So if you want to move across any of these volumes, you could use these scripts, so BigBrain to ICBM and vice versa. And if you want to move across volume spaces, you could use BigBrain to FS average and vice versa. And just to point out, at the moment, what really the engine of these transformations are that we're using is one really important one here is the volumetric transformations from Zhao et al. 2019. Oh, went too far. And so this is a slight improvement on the already registration of BigBrain to BigBrain Sim. This takes it a step further, where they optimized it using manually labeled subcortical regions. So you can see some really important advantages here. So if we just look at, for example, the orientation of the hippocampus, in the yellow one here, this is in the standard imaging atlas. And then you can see it in this new registered version of BigBrain, and its orientation is more aligned to the MNI 152 than just using BigBrain Sim. So this was really optimized for deeper regions in the brain. And so you can imagine for different type of analysis strategies, we might need to use different types of registration matrices and transformations. So we'll also try to keep BigBrain Warp growing so that you could have the choice to use different transformations as they become available as well. And for the surface-based transformations, at the moment, we're just using a really simple approach where there's an identity relationship that already exists between BigBrain and BigBrain Sim, so that's solved. And then from BigBrain Sim to FS average, we just use a nearest neighbor interpolation here because they're in a very similar space already. So you can imagine having a value for a vertex here on BigBrain, and then it's going to be directly mapped to that same vertex or a matched vertex in FS average 5. So in terms of what's in the toolbox, it's all on GitHub. I'll show you a bit more of that in a moment, but the really important parts of this GitHub repository are the scripts and the spaces. So the scripts, there's a few scripts in there, but as I mentioned before, these wrapper shell scripts, bash scripts are really the most helpful part, I think. So this is for moving from BigBrain to FS average, BigBrain to ICBM, or vice versa. And there's a few different options in there. For example, we have them kind of already built for if you just want to use a direct Python or MATLAB implementation as well. And the spaces are currently split up into BigBrain, BigBrain Sim, FS average 5, and ICBM. And so within that, you'll see some maps that we've already pre-computed. So for example, in BigBrain, you'll have both volumetric and surface-based maps. So here you have a histological gradient, so hist G1. We also have func G1, and that has a BigBrain tag at the end. So when you see that tag at the end, it means that it's been transformed from a different space to BigBrain. And you can see that going the other way, if we look at FS average, we created the functional gradients on FS average. They don't have a tag there, but we transform the histological gradients to FS average. They have this extra tag here saying they've been transformed to FS average 5. So BigBrain Warp is really designed in a modular way to support the integration of more techniques as they become available too. And certain transformations I mentioned are likely to work better in certain situations. So we'd like this ecosystem to keep growing, and that really depends on your input. So I'd like to invite anyone who's interested to join the team. If you see something you think you can help with, get in touch. We're more than happy to add more collaborators. Also, if you've conducted research on BigBrain that you'd like to share or incorporate, or even if you'd just like us to direct BigBrain Warp users to it, please let us know. And finally, the current repository is really based on what our needs have been. So if there are any features or options that you'd like to see added, please get in touch and we'll try our best to make that a reality too. And you can find a lot more information at the Read the Docs and on the GitHub. So before I move to the more interactive section, I just want to say thank you to everyone involved from the Montreal side and the Jülich side where I am right now, and across everywhere else as well. So thank you to everyone. So that takes us towards getting started. If anyone wants to follow along now or just for the future, there's two ways that you can really use BigBrain Warp. One is to use it as a GitHub repository. So you just clone the GitHub. That comes with dependencies though. The main one is Mink2. Also there's a few Python libraries, but those are pretty simple ones. And for visualization, I've often run the tutorials using FSL and FreeSurfer, but it is just for visualization. However, if you have any problem with the dependencies, Tristan was really kind in setting up a Docker image for BigBrain Warp, and so now this is working really well. And so if you already know how to use Docker or you want to get to know how to use Docker, you can also use that version of the installation instead. So I'm just going to stop that for a moment. There was a lot of things popping up in the chat. I'm sorry if I missed things, but I hope it's okay. I hope Boris handled the chat while I was talking.
Well, more or less. There's like one question from Lindsay about the registrations between the surfaces.
Yeah,
and I'm
at
sure.
the moment, Yeah, I can. At the moment, the registration between BigBrain and FS Average 5, it's a nearest neighbor interpolation from the mid-surface of BigBrain Sim to FS Average 5. We perform that within each hemisphere separately. It's not perfect by any means, but it's working for us well enough at the moment. But I think that, I know you're working on things, Lindsay, that are more advanced than that approach. So it'd be really nice to kind of, as better techniques become available, we can start integrating them in BigBrain Warp as well.
Yeah, that's already it for the open questions, but maybe
super.
If some more attendees have other questions, that's maybe like a good moment.
Yeah, otherwise I'm going to share a different screen. Okay. So now if anyone would like to follow along with a little test run of BigBrain Warp, you can do so. I'm going to go through a little demonstration based on the GitHub installation. So to follow along, you need those dependencies I mentioned, and you need to pull the GitHub. If you already set this up, just pull the GitHub repo again, because I added this script in this morning. You'll find it in the scripts folder. It's called demo git-based, I believe. And so you'll have the whole code there, and you can follow along if you like. All the data is already contained in the GitHub. And if you're using the Docker version, well, I think it's quite straightforward to translate if you already know a little bit about Docker. If not, then feel free to come back to it at another time, because the script is going to be available in the GitHub, and you'll have this video to follow as well. Cool. So if there's no questions, I can just start talking you through a little bit more of how to use the toolbox. Okay, just to orient you in terms of my screen. So the top is we have this script. So it's the demo git-based. It's all bash version. And then at the bottom, I have my terminal. So you'll see that I'm basically running through the top lines and printing them out the bottom. I couldn't get Zoom to work on my Ubuntu system, so it's going to be a little bit of a, yeah. Okay, so anyway, so first thing we do is we just want to set up our BigBrain Warp directory. So you just want to say where the GitHub repository is positioned. And also set up a working directory. This is just somewhere that I'm going to output all the files to. Then we want to change into our BigBrain Walk directory and just pull the latest version of the GitHub. And then this is kind of an important part. If you're using the GitHub-based installation, you need to go into this initialization script and just change the first two lines. So it needs to know where your GitHub repository is located, and it needs to know where Mink is installed as well. So just the first two lines of that script need to be changed depending on your system. That's not necessary if you're using the Docker version though. Then you want to run those scripts. So you want to export those paths essentially, and also run the scripts. It'll also download a bunch of dependencies that you need, some of the transformation matrices, different things from OSF that are too large to fit on the GitHub. And then we're ready to go. So the first thing we're going to try out is a surface-based transformation. So here you can see the line I'm highlighting. So we're going to move something from BigBrain to FS average. Specifically, we're going to move a histological gradient from BigBrain to FS average 5. So we have two inputs. They are the gradient values on the left hemisphere and the gradient values on the right hemisphere. You can put this in as a text file, various types of FreeSurfer type formats like an anot or a curve file, or also works with Gifty. So you just put in the left data, the right data, and then the last thing is just where it's going to output that data to. It'll append this with underscore LH underscore FS average 5.text. So it just appends it with the hemisphere and then the surface that you're transforming it to, and it just outputs it as a text file at the moment. And it will do the same for the right hemisphere. Okay, so that takes a couple of seconds to run. It's really straightforward at the moment. I just want to check, is there anything in the chat? Okay, I don't think there's anything I need to reply to right now. Okay, so
Casey, there's just one question,
yeah,
but it's not related to these lines of code. It's just like, how do you get cortical depths from quantitative T1 images or from T1 weighted images? Is that possible using MPC approaches, for example?
yeah.
And how do you relate this to BigBrain?
Right. So for the approach in structural imaging, so Conrad will talk a lot more about construction of equivolumetric surfaces later, I believe, but as a brief overview, you essentially take your peel surface and your white matter surface from, for example, FreeSurfer, and then you construct these equivolumetric surfaces between it. So it's a set of surfaces within the cortical mantle. And then you can use different commands. I'll go through one in a moment called volume object evaluate, for example, which is a min command where you sample the intensities along that surface from a volume. So you could use a quantitative T1 scan or a T1 over T2 scan. They just need to be aligned to your surface space, and you have a set of surfaces that you sample along. And then you have fewer surfaces than you would for BigBrain because it's much lower resolution. But we have seen that there's quite a good concordance of the gradients that you can obtain from this. There's still some really interesting features you can obtain even from lower resolution MRI data. I hope that answers the question. So with that, we'll go back to the coding. So here we can now visualize this transform data. Oh, I need to play. To do so, we need to make a few things FreeSurfer compatible. So that involves transforming some object files, which are a type of MINC format for surface meshes, to a FreeSurfer format. And we also need to convert the text to a curve format. So this is really just some transformations to get things into a FreeSurfer form, but this is really just for visualization. And then we can look at these histological gradients now on a different array of surfaces. Here it is on the histological BigBrain surface. So this is where it was originally constructed. So you can just see a few noticeable things that you have really the peak of the gradient here on somatosensory and visual areas, for example. Then we could change the BigBrain sim surface. This already has this identity relationship, so it's quite a simple one. And then finally, we have the FS average transformation. So we can kind of check out if the transformation has worked well by just kind of comparing the values at different different points in the cortex, for example. So we see really high values right in these deep primary sensory areas. So we have a decent alignment between BigBrain sim and FS average 5 there that you see. Okay, so that's a surface-based transformation, just one line of code. I think, is there another question? Okay, how easy would it be to apply BigBrain Walk to non-FreeSurfer generated format surfaces? So at the moment, we only have it for FS average 5, but I've contained the code in the toolbox that I use to do that mapping. And we could repeat it, or if anyone's eager, they can also repeat it for different types of surfaces, as long as they're in the same space. If they're in MNI 152 space, then you could also just try doing a nearest neighbor interpolation, which in the BigBrain Walk toolbox, there's, I think it's called nnsurfaceindexing.m, which is a MATLAB script that I use to do it. And so in practice, I think it'd be quite straightforward to do it for any surfaces that are already in MNI 152 space. And we plan to do it for example, the Conti 69 surface pretty soon as well.
Yeah, cool. just like a follow-up, like for Conti 69 and FS average, you can also use Workbench to map between the two surface spaces. So that could be used in addition.
And then Nicole just asked, how is a script like BigBrain to ICBM different from writing a registration warp directly? So at the moment, I'll show you actually, when we get up to the, well, we're about up to the volumetric one here. So I can talk you through kind of what are the components of this. So here we're just going to look at, for example, let's take a motor activation. This is from Neuroscience. So this is just in MNI 152 space. We can check it out. It looks like a typical motor task type activation. So this is the ICBM to BigBrain script. So here, the input is going to be that Nifty file. It can also be a Minc file though, just something that's aligned to MNI 152. Then the other arguments we're going to use are histological or sim. So which BigBrain space do we want to go to or come from? So those are the two options at the moment. And then we can also choose whether we want to do a linear or nearest neighbor interpolation. So the inputs there would be either linear or nearest, and then you say the output. So to answer Nicole's question, at the moment, this is very similar to if anyone knows how to use Minc resample. It's just a wrapper script for Minc resample, and based on these options that you give it, we kind of change the options of the Minc resample tool. And we would hope that in the future that we can make this more flexible, that if there's other volumetric transformations, there could just be another argument in here. So there's just essentially it's all wrapped up, so you don't have to know how to use Minc tools, for example, to do this. And especially as we start using the Docker more, I think that could be really helpful for people who aren't acquainted with Minc. Was there a follow-up? Cool. Okay. So this one takes a little bit longer than Minc resample. So let's just fast forward a little bit. Okay. So now it's finished resampling this motor activation to histological BigBrain space. So let's have a look at how it's performed. Okay, looks decent, right? It looks like it's transformed quite nicely over to that histological space. Okay, I think there was another question. Oh, good. Okay. So I think one of the real advantages of using BigBrain, at least personally, is using the surface data, because you could use these staining intensity profiles. So even if you had this, for example, task activations, you've moved it to the histological space, the next step might be to go from the volumetric space to the surface space of BigBrain. So we can do this pretty straightforwardly using some Minc tools as well. So here we have, so here we're going to convert our output to a Minc, depending on what your input is, by the way, to things like ICBM to BigBrains, these volumetric transformations, it will mirror that as the output. So you can either put in a Nifty or a Minc and you'll get the same format back. So here we put in a Nifty, so we get a Nifty back. So we're just going to change it actually to a Minc, so we can use Minc tools. Then we use average surfaces to create a mid-surface of BigBrain. There are actually maybe some other mid-surface models that you could use as well. You might need to test out what works best depending on how much your volume kind of covers the intracortical area. But here we're just going to use a mid-surface that's an average between the gray and the white matter surface. Then we're going to transform this to a FreeSurfer format, just so we can view it once more in free view. And then we'll use this really handy volume object evaluate tool. I mentioned this before. You put in a volume and you put in a surface, and it's going to sample the intensities along that surface within the volume. Okay. Were there any other questions? So I think Timo answered one for us. Okay, so now let's see how this looks on the BigBrain surface. Cool, so now we have our histological BigBrain surface, and we're going to have a little motor activation now on the surface. So you can see there are some points that maybe you need to check the surface registration as well to see whether this is due to like the volume not covering the whole cortical surface. So these are things you have to watch out for as you're doing the sampling between volume and surface space. But in theory, we really do have a pipeline that at least we could test these things quite practically. And then you could perform some type of investigation of cytoarchitecture within these areas that are activated during a motor task. Okay, I think I have just, do I have, what time is it? Oh, good, I still have a few minutes left. So let's look at one last example. This one's super quick, just because I want to point out you can also do this with different types of input data for the surface spaces. So you could also put in a .anot file, so a parcellation scheme from FS average, and then we can move it over to BigBrain space. So let's try this with the 7 networks. So we put in our annotation files, just left hemisphere, right hemisphere, then how you want to output it. We do a little conversion just to make it viewable with Freeview. And now we'll have these networks on the BigBrain histological surface. And once again, we could then look at the cytoarchitectural differences across different functional networks. Okay, so that's the end of our little run through. As you see, the scripts are pretty straightforward. And the idea will be that we can add more potential approaches to these, so you can test them out for yourself as well, depending on what your analysis strategy is. So if there's no more questions for the moment, I can also take some time to just show you the website. Oh, there's one in the chat. Oh, thanks, Sophie. Okay, let's. Cool. Okay, so this is the BigBrain Warp Read the Docs. Hopefully this covers even more actually than what I showed today. So I'll just take you through a few things that we haven't discussed today. So installation we kind of discussed. Then we'll have a little bit more on the background of BigBrain, kind of some intuitive things that we've learned over the years of working with BigBrain that might be helpful to you, and also some resources you can find. You'll find what's inside the toolbox here. And maybe I'll also just highlight that these are the types of tutorials that we've invested some time into making. So it's really talking through how we go about these analyses to get your head around like all the problems that you might face as you do these BigBrain MRI analyses, and guide you through step by step what our studies have really entailed in terms of relating cytoarchitecture to function. And it also gives you a bit more background into how we've created the toolbox as well. So feel free to give those a read if you're interested in performing these analyses. Another thing is we're trying to use this as a way to point people in the direction of other research that helps link between cytoarchitecture and function. So you'll see here different links out, for example, to eBrains, where you can find these really precise cytoarchitectural areas related to Jülich brain as well, and a couple of other things such as like the layer segmentation that Conrad will show later, the hippocampal segmentation that Jordan performed, and on the in vivo imaging side, we'll also link out to other toolboxes that could be really nicely integrated with BigBrain, but maybe haven't been yet. And then otherwise, it's just, feel free to ask us questions and get in touch as much as you like. But I think that about covers it. Hi, Alan.
Hi, Casey. And this is a fantastic presentation, but one of the questions that often comes up is the BigBrain is a single individual. And when we're mapping it into MNI 152 space or any other standard space, that's a different brain, or a composite of brains. Could you speak a little bit about how one can overcome the individual to average mapping function? BigBrain has one Heschel's gyrus, and the average has 2 Heschel's gyrus. How do we deal with that issue?
Yeah, I think for a large part, the way that we've dealt with that is really closely inspecting the surface, like for example, surface to surface mapping at each occasion. For example, when I was presenting the project on the mesotemporal lobe, we started off using both hemispheres, and then we look closer and closer and find that there's actually a little tear in the entorhinal cortex of BigBrain. So then we have to reevaluate our analysis strategy. So I think that for us, it's really been about having detailed look at BigBrain for each occasion, and understanding that it's not going to be a one size fits all in terms of performing some small region of interest mapping. to whole brain mapping, and really having to take that into account in your analysis strategy. I would say it's really, you have to consider it for each project independently. But I think that another thing that we've tried to do is validate it with quantitative imaging as well. And so that's why we've really tried to build in these similar approaches with MRI to see if we can find similar types of patterns using large cohorts of individuals where we have matched functional and structural imaging data, but we lose out on spatial resolution. But I wouldn't say the problem is solved, but I think one hope is that by using BigBrain Warp, we can start to compare different types of surface to surface mapping or volumetric mapping to see the accuracy and to see what the advantages and drawbacks of different approaches would be, because we now have a really easy to use tool to do so.
Thank you.
If I can just follow up on this question with like one thing, I think, you know, for the Chiao paper that is like this advanced BigBrain sim space, I think what was key there was, I mean, what you say is that the imaging was kind of not conventional imaging in that sense, right? It's like this T2 weighted imaging, for example, where the template was built. So this was possibly closer to BigBrain contrast, and then you could kind of maybe improve the mapping to go to MNI 152 space by having this intermediary PD25 space. So I guess if we can kind of build good intermediary features, then maybe the registrations can successfully be improved.